A single molecule of glucose holds an immense amount of potential chemical energy, but it doesn't give that energy up easily. Its covalent bonds are extremely stable, locking that power away inside a rigid 6-carbon ring. Breaking every bond in that molecule at once would release nearly 700 kilocalories per mole, an amount of heat that would be unmanageable for a microscopic cell. To avoid this, cellular respiration performs a controlled oxidation, harvesting that energy in small, manageable increments. This extraction process begins in the fluid of the cell's cytosol, strictly outside the specialized machinery of the mitochondria. As this diagram shows, the initial pathway occurs on the far left, glycolysis, the splitting of sugar. The process starts with one 6-carbon glucose molecule, systematically broken down into two 3-carbon pyruvate molecules, shown in these yellow boxes. However, because glucose is so chemically stable, the cell faces a problem. It must initially operate at a deficit, spending its own energy reserves just to start the process of breaking those bonds apart. To manage this, glycolysis is divided into two distinct halves, an energy investment phase, followed by an energy payoff phase. This upfront cost is a biochemical necessity. Spending energy is the only way to destabilize the sugar enough to force it to yield a much larger return later in the cycle. In the energy investment phase, we can track exactly where the cell's ATP is spent to dismantle the 6-carbon structure. In step 1, the enzyme hexokinase uses ATP to add a phosphate group to glucose, forming glucose 6-phosphate. In step 2, it rearranges into fructose 6-phosphate. This shift from a 6-sided to a 5-sided ring prepares the molecule for further destabilization. Then, in step 3, a second ATP molecule is spent by the enzyme phosphofructokinase. It adds another phosphate to the opposite side of the ring, creating fructose 1,6-bisphosphate. Attaching these two negatively charged phosphate groups creates immense structural tension. The molecule is now highly unstable and primed for fracture. That tension is released in step 4, where the enzyme aldolase cleaves the unstable 6-carbon molecule directly in half. This cleavage produces two distinct 3-carbon molecules, dihydroxyacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate, known as G3P. Only the G3P molecule can proceed directly to the next phase of respiration. Because of this, an isomerase enzyme in step 5 rapidly converts the DHAP into a second molecule of G3P. From this point forward, every single reaction will occur twice for every one molecule of glucose that entered the pathway. By spending 2 ATP, the cell has essentially set a molecular trap. It has irreversibly committed the glucose to breakdown and doubled its potential return by creating 2 reactive G3P molecules. The cell now enters the energy payoff phase, where it will recoup its investment by stripping the G3P molecules of their high-energy electrons. Step 6 is the critical oxidation event. We can see the G3P molecule being oxidized, losing electrons that are immediately captured by a carrier. This carrier, NAD+, acts as a shuttle, capturing these extracted electrons to become NADH.
Because we have 2 G3PP molecules, this step generates 2 molecules of NADH. In step 7, the cell performs substrate-level phosphorylation. A phosphate group is transferred directly from the intermediate molecule to ADP, forming ATP. Since this happens twice, this step alone produces 2 ATP. At this moment, the cell has exactly recovered its initial investment. Steps 8 and 9 involve internal rearrangements and the removal of water to create a high-energy compound called phosphoenol pyruvate. In the final step, step 10, the enzyme pyruvate kinase harvests the remaining phosphate groups to generate more ATP. The final tally for the payoff phase is 4 ATP. After accounting for the 2 ATP spent in the investment phase, the cell is left with a net gain of 2 ATP per glucose. The remaining carbon skeleton at the end of this 10-step sequence consists of 2 molecules of the 3-carbon compound pyruvate. In just 10 cytosolic steps, the cell has successfully secured a net profit of ATP and NADH. It has extracted this energy solely through the rearrangement of sugar bonds, without requiring a single molecule of oxygen. If oxygen is present, the process moves deeper. The 2 pyruvate molecules leave the cytosol and cross the double membrane into the mitochondrion. Here, they undergo the link reaction. Pyruvate cannot be used by mitochondrial enzymes in its current 3-carbon form.
it must be modified to proceed. As the pyruvate enters, the first modification is decarboxylation. The carboxyl group is snipped off and released as carbon dioxide. This marks the first time carbon from the original glucose molecule is fully oxidized and expelled as waste gas. Next, the remaining 2-carbon fragment is oxidized, transferring another load of high-energy electrons to reduce NAD+ into NADH. Finally, a sulfur-containing compound called coenzyme A is attached to that 2-carbon fragment. This creates acetyl-CoA, a highly reactive molecule specifically designed to deliver its 2-carbon payload into the next phase of respiration. The link reaction effectively serves as a mitochondrial checkpoint. It exacts a carbon tax in the form of CO2 in exchange for producing the specific 2-carbon fuel the mitochondria require. To understand the progress made, we need a strict accounting of what has been gained from that single original glucose molecule across both glycolysis and the link reaction. We have a direct net gain of 2 ATP, all generated in the cytosol. We have 4 molecules of the electron carrier NADH, 2 from the payoff phase and 2 from the link reaction. We have released 2 molecules of CO2 as carbon waste. And we are left with 2 reactive molecules of acetyl-CoA sitting inside the mitochondrion. When we look at the full map of respiration, we can see where these resources are headed. The NADH is being channeled toward the electron transport chain on the right, while the acetyl-CoA drops into the Krebs cycle. But 2 ATP is barely enough to sustain cellular life. The vast majority of the energy from the original glucose remains locked inside the covalent bonds of acetyl-CoA and the electrons held by NADH. To harvest that remaining potential, the cell must now initiate the Krebs cycle.